thanks to the organizers to be part of this great meeting among these great speakers, and to present some of our work on voltage-gated calcium channels and some regulation mechanisms by post-translational modifications. So just a brief overview. The excitation-evoked calcium influx is important for pretty much every excitable tissue. It regulates variety of different processes, neurotransmitter release, excitation contraction coupling in skeletal muscle and heart. It can also trigger gene expression alterations. And voltage-gated calcium channels are responsible for this regulation of these processes because they basically respond to membrane depolarization by allowing calcium entry. And they are formed by these large molecular complexes. Basically, there is a main subunit called alpha-1 subunit traditionally, and it has a property to form a calcium selective pore and a voltage sensor. So basically that regulates the pore. This is highly schematic how it looks like, and there are 10 mammalian genes encoding voltage-gated calcium channel pore subunits, basically classified in these three major families. And all of these require co-assembly with auxiliary subunits, auxiliary proteins, to form properly functional channels, apart from CAV3 subgroup, which can function on its own. Basically, this talk is mostly about N-type calcium channels, which are also called CAV 2.2, and they are very important in the presynaptic terminal of sensory neurons, but also in the central nervous system. And in the case of N-type channels, the auxiliary subunits are cytoplasmic beta subunit that helps the trafficking of the channel complex to the plasma membrane and modulates some functional properties. And also extracellular alpha-2 delta subunit, which is basically the main focus of this work. And the function of alpha-2 delta has been somehow not well understood, basically, because it is a very, this is what I'm showing is a recent cryo-EM structure which showed this protein is the skeletal muscle version of the calcium channel as in all its beauty. So basically you have alpha-2 delta subunit entirely extracellularly interacting only with the channel at the exofacial interface. So the function of this subunit has been difficult to understand because it is very challenging biochemically. And basically, just several more words about alpha-2 delta. There are 4 mammalian genes encoding 4 different alpha-2 delta subunits. And alpha-2 delta 1 and 2 subtypes have been very interesting for the pharmaceutical industry because they are the main target for binding of gabapentin, which is prescribed against chronic pain. And then in terms of function, basically in heterologous system, it's known, very well known, that alpha-2 delta subunits increase the current density of CAV 1 and 2 subtype of calcium channels. This is illustrated here, so different depolarizing potentials. The currents are bigger in the presence of co-expressed alpha-2 delta. This is data from heterologous cell lines expressing the calcium channel subunits, CAV 2.2 beta. In the absence of alpha-2 delta, there is less current. It's shown here in this IV plot where the depolarizing steps are plotted against current density. So this can be due to more channel protein expressed at the cell surface or to some changes in open probability. And exactly this is kind of controversial, but it's well known that this is partially due to increased surface expression. Well, there is different lab has shown that basically co-expression of alpha-2 delta subunit increase the channel protein, but expressed at the cell surface. But this is not proportional to the huge current increase, and there are also very likely other mechanisms involved. And another thing that is worth to know that alpha-2 delta is actually highly post-translationally modified protein. So it's glycosylated. It has actually a C-terminal which is being cleaved off and replaced by GPI anchor. This is, our lab was the first to show that mechanism. And also it's post-translationally cleaved into alpha-2 and delta part and remain associated with disulfide bonds. So you've got this extracellular protein sitting on top of the channel that is modified so heavily. And just one experiment of the long time ago when I first started in the lab of Annette Dauphin, when I basically was able to remove the GP anchor basically by using this enzyme, PLC, which chops off the hydrophobic part of the protein, which is GP anchor, and we were able to show that the protein actually redistributes in the from hydrophobic phase, detergent enriched phase, enriched in hydrophobic proteins, into hydrophilic aqueous phase. So it's by this phase separation assay was one of the evidence that went into this paper a long time ago, showing that the protein has actually the GPA anchor, which was afterwards actually confirmed in these cryo-EM structures, happily. So I'm not going to talk about this today, just about, I'm going to focus on another property, which is the post-translational cleavage of the subunit. So as I said, it's being cleaved by yet an unidentified protease, but and it's been known for a long time that the protein is cleaved. Since the channels were first purified from skeletal muscle, the Bill Cottrell group has found that there are several bands, which they called at the time alpha-2 and delta. Up to now, they're called like this. And then they have shown that alpha-2 and delta are actually basically encoded by the one gene, which is post-translationally cleaved. But since then, the functional role or the protease involved was not well understood. And my project for a long time was what is the function of the proteolytic maturation of alpha-2 delta in the context of calcium channels. And basically, we didn't know the protease. We still don't know which protease cleaves the protein, but we know the proteolytic site that the protein is being cut by the delta peptide has been sequenced. And it's a sequence that actually reads cleave me, which is kind of suggestive for the site. But we couldn't, the way to manipulate the sequence was to replace it with a target site for a exogenous protease. In this way, we could manipulate the proteolytic cleavage and study. So we chose viral protease called 3C protease from human rhinovirus, which we could co-express as a cDNA, and we replaced the proteolytic site, calling this construct 3C alpha-2 delta. Luckily, that prevented the proteolytic cleavage by the endogenous protease. This is data from heterologously expressing TSA cells. So there is only one band expressed at the plasma membrane, which is this is biotin-related material. And then we quantified in many times repeated this, and also by immunocytochemistry. Just the idea of these experiments was to show that the cleavage did not affect the plasma membrane expression of the subunit. So the protein was expressed at the membrane but was not proteolytically cleaved. And then we tested the functional significance of that. We co-expressed the other calcium channel subunits, CAV2.2 and beta. And basically, the effect of alpha-2 delta subunit to potentiate the calcium currents was completely lost, like to zero, pretty much, in this 3C alpha-2 delta constructs with manipulated proteolytic site. It's shown in the IV curves here. So it's pretty much as in the absence of alpha-2 delta subunit. So the functional effect was lost. Then to see whether this will affect in any way the plasma membrane expression, we used a bungarotoxin tagged alpha-1 subunit, which had the BBS tag at the extracellular site, and we could label in live cells with fluorescently labeled bungarotoxin, the surface channels. An example is shown here. These are the cells labeled with CAV2.2 overexpressing the channel subunits in all these conditions. And interestingly, we found that, as it was shown before, alpha-2 delta increased the surface expression, relative surface expression, about 2.5-fold, but a 3C alpha-2 delta also did increase. So the effect on plasma membrane expression wasn't abolished. So it's not an effect on surface expression then. So next thing we did was to co-express 3C protease with the 3C alpha-2 delta to induce cleavage artificially and to see whether this will affect the function. And first we checked if this will work. It did, so that's the protease was co-expressed as a cDNA. So basically this is a plasma membrane isolated protein. It was cleaved by the protease at the inserted site. And then this led to restoration of function. Basically, this corresponds to lack of protease. And then this, I mean, CAV2.2 channels were in beta 1b and 3C alpha-2 delta were expressed here in the absence and with the protease. Or that's an inactive version of the protease, which has a mutation in the active site. So the restoration of function was partial. We had, but significant. It's shown here in the IV plot. So basically, the channels, the function was restored. So this is a scheme I made, which how we imagine things are happening. So. The trafficking in these heterologous cell lines expressing the channels seem not to be affected, the surface expression seem not to be affected by the proteolytic cleavage. So, but we showed that the channels are expressed at the cell surface, but they cannot be activated by voltage in the absence of cleavage. And then we need to induce cleavage by co-expressing the protease, and then we end up with channels that can be voltage activatable. So if this is true, we were really keen to try extracellularly to apply the protease in live cells. And for that, I had to change the strategy a little bit and use another alpha-2 delta subtype. I was talking about alpha-2 delta 1 until now. This is alpha-2 delta 3. But you have to believe me, alpha-2 delta 3 has exactly very identical properties to CAV2.2 channels to alpha-2 delta 1. So I replaced the endogenous proteolytic site of alpha-2 delta 3 with a site for the protease thrombin, exogenous protease. And then the idea was to apply thrombin in cells. This was done for technical reasons. Instead of 3C protease, I used thrombin because it just worked. So then I tested if this treatment with thrombin in cell culture will induce cleavage, and it did. So the alpha-2 delta with the thrombin site, non-treated and treated for 1 hour with thrombin. This is from the plasma membrane biotinylated fraction. The cleavage was present, and it was at the inserted site, and then this restored the function again. So this was additional evidence that the channels require this proteolytic cleavage to be allowed to be voltage sensitive. So that was very interesting. There was also the shift of hyperpolarized potentials, which is also one of the effects of alpha-2 delta that appeared. So basically, this is what the currents correspond to. So this proteolytic cleavage restored the functional effect of alpha-2 delta. And to summarize the data in heterologous cells, we found that proteolytic cleavage enables the functional effect of alpha-2 delta on CAV2.2 currents. And the mechanism was independent of channel trafficking, or to sort of say to cell surface expressed channels in heterologous cells. And just I have 3 or 4 slides at the end to show what we saw when we express these constructs in primary neurons. So we used, the question we asked was does the proteolytic maturation will affect CAV 2.2 channel trafficking in transfected primary neurons? We used hippocampal neurons from P0 rats, and we expressed CAV 2.2 channels with an HA tag, beta 1b, and alpha 2 delta subunits, and M cherry as a marker for neuronal morphology. And basically, in the presence of alpha 2 delta, the channels were all over the processing of primary neurons. When alpha 2 delta was not co-transfected, basically the channels didn't traffic at all. And in the presence of 3C alpha 2 delta, we also saw exactly a very similar situation. There was no sort of trafficking of the subunits, channel subunits, and then co-expressing the protease did clearly restore that. So we saw basically the channels could be trafficked again along neuronal processes. There's just a quantification of many experiments showing that alpha 2 delta, basically what we quantified was the amount of CAV 2.2 in processes at certain distance from the cell body. And basically in the presence, absence, alpha 2 delta or 3C alpha 2 delta. And then this is a restoration effect by the protease. So artificially we could sort of show that this proteolytic cleavage is very important for also trafficking in neurons, and it was kind of contradictory to what we saw. I'm sorry, I switch between slides. So it was kind of contradictory to what we found in heterologous cell lines like TSA or N2A cells. And the rationale to explain it is that it's well known that in neurons that are more sophisticated mechanism for control of trafficking, and there's a more quality control checkpoint mechanisms. So we think that in not a correct conformation, the channels associated with uncleaved alpha 2 delta are simply not trafficked. One explanation is that also only active channels would go to the right place. So yeah, these are again schemes I did to explain what we found, which are very simplistic, but yeah. And one last slide is just to show where this, we have some data showing where the cleavage occurs in cell lines subcellularly, but I'm not going to have time to go through that. Just show you this experiment, which is from in dorsal root ganglia neuron. Express high levels of alpha 2 delta 1. It's actually one of the genes that is upregulated in neuropathic pain states. And this is isolated from a spinal nerve ligation model of such neuropathic induced rats. And basically in these states, alpha 2 delta is highly upregulated, and we see a lot of this uncleaved form, which is present only at the cell body of these neurons. But when we go along neural processes in both sides, we see only proteolytically cleaved form, which is shown here. So it's again an indication that during the trafficking somewhere, the proteolytic cleavage occurs. And we also did some experiments overexpressing this uncleavable version of the subunit just in DRG neurons. And there's a preliminary actually results, but we saw an inhibition of endogenous currents, suggestive that endogenous channels could also actually be inhibited by this subunit. So to summarize, alpha 2 delta proteolytic maturation is important for enabling the voltage-dependent activation of CAV 2.2 channels, and to enable the trafficking of CAV 2.2 complexes in primary neurons. Most of this work was published in eLife last year. Inhibitory role of pro-alpha to delta could be present by recruiting endogenous channels. And also it's very interesting to look into which protease is involved, because that could be a target in regulating the calcium channels. And because alpha to delta is associated with all the L and N and PQ type, etc., calcium channels required for their regulation. This is the lab photo. This was done in the lab of Annette Dauphine, and all the people that worked on this are also listed. And thank you for your attention.
Some questions for Ivan?
Nice talk, Ivan. So you have done these studies to determine that it's not necessarily a trafficking effect by the proteolytic cleavage, certainly in your heterologous expression. Have you done any single channel recordings to determine PO?
No, I haven't.
And then, so the next question would be, you've alluded to several times that it's enabling voltage dependence of activation. Have you got data to suggest?
Basically, no. Probably I shouldn't say that, but this is but this is what we think. I should have said that this is what we think is happening. I have no, I don't have data of looking at gating currents and looking at if that will be blocked. We haven't done that. But I'm working further on that, and I would like to look into like the effect on the gating currents and actually the voltage sensor. And
that was almost my next question. Is so do you think there are additional contacts between alpha 2 delta when it's cleaved and the voltage sensor potentially that might?
Actually, I went to a lab that I went for 3 months in the lab of Riccardo Cese in UCLA to look into the effect of voltage sensor on the voltage sensor, and I have some data on that which I can't show yet, but it doesn't. It's not so easy to answer this question yet.
Okay, thank you.
I would like to know. Yeah.
Hi, that was really interesting.
Thank you.
One question I have is, do you think, other than a sort of pathophysiological modulation effect, this could be more sort of developmental as well and more fundamental to the overall expression pattern of these channels?
Well, there are different possibilities, and that's one of them. What I find interesting as an explanation is that could be a general mechanism to keep these channels closed until they need to actually they are needed. Because once they are in the endoplasmic reticulum, there is very high calcium, for example.
Yeah, they're very fundamental, aren't they? Yeah.
They could be somehow a way to keep them inactive, which will be difficult to show. But it could be developmentally. We haven't really monitored developmental cleavage effect, and we don't have enough results on subcellular location of cleavage yet.
Okay, thank you.
Yeah.
Am I right in remembering that some calcium channels are cleaved also in the C-terminal?
Yeah.
And that also modulates channel activity. So what do you think of the way these two proteolytic mechanisms might be interacting in modulating the channel overall?
I think they're completely, my interpretation, they're completely independent of each other, because this is entirely outside. And we actually think that it could be also happening at the plasma membrane. I mean, it's not completely excluded that the protease is extracellular in cell matrix. But this C-terminus of CAV 1.3 is cleaved off, and I think it's calpain the protease. And it inhibits the channels, yes. So that's another mechanism also involving it.
I was just wondering what would happen if you cleave the disulfide bond as well.
We did try, I did some experiments trying to reduce to apply reducing reagents. not very successful in my hands. But it's an interesting question.